For this next part, we're going to take the next step and looking at human in vivo brain imaging and linking some of the genes that Dan has just talked about with some of what we are learning in brain imaging. And I'm going to focus particularly on this concept of connectivity that many of you have heard about, but appears increasingly to be a critical issue in autism. So what do we mean by connectivity? Brain development is defined by establishing functional connections in between neurons, like the neurons I've shown here. And initially what we find is that neurons early in life grow. They start small and they grow many processes. They start sending connections all over the place. And some of these connections find good targets and some of them don't. And the functional connections, those connections that line up, they hook up, they work well, they become reinforced over time, they become strengthened, and ultimately non-functional connections are pruned, as we say. They become less and less present. And so there's this process of elaboration and then pruning of functional connections. As Dan showed you before, many, if not most, of the autism risk genes that have been identified thus far relate to some aspect of neural development and establishing connections and communicating between neurons. And so we've got good reason to believe that this is a great place to start in understanding brain and autism. So I like to use this analogy of trying to get from the Valley down to UCLA and thinking about all the many routes that you could possibly take if you're coming from someplace like Valley Village. So imagine, I live, actually I live in deepest Calabasas, but I have to go through the valley. So imagine here you are in the valley trying to get down to UCLA, and there are oh so many routes that you could take, right? You could stumble along here and eventually move your way over to Roscommare or Beverly Glen, or you could go down Beverly Glen or Laurel Canyon or Coldwater Canyon and eventually come over Sunset. If we lived in a rational world where freeways were actually faster than side routes, then what you might do is you might find a big connection like the 405 South, and that would be a nice fat route that you would take. It would be very efficient. Okay, and so over time that connection would be built up and built up. You get more and more lanes there, and these other little connections, these little side routes, would be less and less used and closed off and die off. And that's sort of how it is in the human brain as well. This is an image of brain and myelin. So myelin is that fatty covering over white matter that forms the connections, and myelin shows is what happens when connections, it makes it possible for these connections to move very quickly from neuron to neuron. And this shows development from 107 days to 329 days. It's studied by Dione et al. Just recently, and the red below it shows the pattern of myelination. And so if you look at this just in a global way, you can see that between 100 days and 300 days of life, which is not very much life, there's this massive increase in white matter, in the development of these white matter tracts and the myelination of these white matter tracts. It's from virtually nothing to this very dense set of connections. So connecting and learning to connect is what all of development is really about. Another way to look at that is to look under a microscope at neurons and the neural connections. So here are neurons, and these long lines are their connections, and here is a slice at birth. There's one at 6 years old, and there's one at 14 years old. So you can tell at the very beginning of life there's not a lot happening there. But then there's this massive proliferation of connections. But over time, what happens is those start pruning down and basically going to the most efficient, to the best possible collection of connections. And this is done in a very patterned way, which I'll talk about in a little bit. So this process is very important, and the timing of it is extremely important. This is what allows us to learn, make connections, figure out what areas of the brain are going to talk to other areas of the brain, and to do it as efficiently as possible. Some studies have been done to suggest that autism can be seen as a connectivity disorder. And when we think about this, we can think about both structural connectivity and functional connectivity. In the grossest sense, structural connectivity may refer to simply the size or the amount of white matter in the brain. The white matter is the part of the brain that is just the connective tissue. The gray matter is where the actual neurons live. And this is an old study by Martha Herbert from Boston, who looked at white matter in individuals with autism and typically developing individuals. And one of the things that she found was that there were some abnormalities in the amount of white matter in different sections of the brain. In particular, in long connections, for example, connections like this that connect the two hemispheres of the brain, the corpus callosum, there was a decrease in the amount of white matter, whereas in some of these sort of more local areas, all connecting within parts of the cortex, there was too much white matter. So it seemed as if there was this abnormal patterning in which the long-range connections, there wasn't as much material there, and there was too much in these local areas. Another way to study this is by studying a different MRI technique called diffusion tensor imaging. And diffusion imaging is simply a way of measuring the white matter tracts of the brain, and these are images. The color coding stands for which direction the fiber tracts are going. And I've got a DTI picture of an adult on the right and a DTI picture of an infant on the left. And I hope that it becomes very clear to you just by looking that there are these tremendously elaborate white matter connections in the adult brain, and there are just a few very fundamental connections, mostly connecting the visual cortex and connecting the motor cortex to the brainstem and down. Very, very basic connections, but lacking all of that tremendous development that occurs. So there's a huge, huge development of white matter that we can measure with this kind of imaging in individuals, and we can do this in infants as well as in adults. Some of you may have read this study that just came out last year from Joe Piven's group, looking at white matter development using this diffusion tensor imaging approach in infant siblings. So infants who are siblings of children with autism who are at increased risk themselves of developing autism. And at the time that these pictures were taken, and the study was published, they still don't know which children are going to turn out to have autism and which ones are not. So this is basically an average of a bunch of children, some of whom are more likely and some of whom are less likely to develop autism. And the study just looked at two different parts of the corpus callosum, this front part here and this middle part over here. And you can see there's a lot of variability across individuals, but the main point is the developmental trajectory as you go from over here at 6 months all the way over here at 27 months. That the kids in blue here are the children who are not from high-risk families, and in red are the kids who are from high-risk families. And you can see two things. One is that these kids from the high-risk families begin with slightly more white matter, and then the development in this corpus callosum region just doesn't develop at that same rate that it's supposed to, whereas in the kids from the low-risk families, as you can see, there's a nice developmental trajectory, and this is true of both areas of the corpus callosum. And this is this is in the first 2 years of life, so most of these children are not even old enough to get a solid diagnosis of autism, and yet we can see that though, and many of them are showing no signs of autism throughout this part of the lifespan, even though it's clear that something must already be happening in the brain in the first 2 years of life. And that's what's very important, and that has guided a lot of the studies that we've been working on and that we're particularly working on right now. Now, the other kind of connectivity we can think about is what we call functional connectivity. So those studies where structural connectivity just shows us the structure of the white matter in the brain. Now we're going to talk about the function of the brain. And in functional connectivity, we can use techniques like functional MRI, where we have individuals go in the scanner and we can either have them do tasks or we can just watch their functional activity at rest. Because areas of the brain that are highly connected will change their activity over time in concert with one another. So for example, these are two different areas in the brain that form part of a network that is called the default mode network. I won't talk about why it's called that, but just to know that these are areas that are known to be part of the same functional network. They work together. And if you look at the time series of those regions, you see this is one region, this is the other region, that as one goes up and goes down, the other one goes with it. So that's what we mean by functional connectivity, two areas of the brain whose activity patterns worked in concert with one another, either positively or negatively, but they form a network, a functional network together. There are lots of ways of hypothesizing functional connectivity deficits in autism, and there have been several models out there. One of the first models was a model of very simple underconnectivity. This was developed by Marcel Just, and this is a graph just showing connectivity in control subjects in children with autism, showing decreased connectivity across a whole range of time epochs that were measured. But I think increasingly it's been thought that the story is more complicated than that. It's not simple underconnectivity. It's that the connectivity is aberrant. It's different. In some areas there might be overconnected. In other areas it might be underconnected. And another popular theory, and one that Dan Geschwin and Jennifer Levitt here, as well as Matthew Belmonte has talked about, is this whole concept of developmental disconnection. That is, the whole developmental trajectory of functional connectivity is impaired. And there are lots of ways you can visualize this. I'll show you some better pictures than this, but you can imagine of different nodes in the brain, different areas of the brain having to communicate with each other. And in this model, you have several different nodes in the brain, little networks that communicate over long ranges in efficient ways, whereas in autism we may not see some of those long range connections, but a lot of overgrowth of these local connections, and that's the model I think that's most popular today. There have been several studies of this coming from particularly Jeff Rudy, who's a former graduate student of Mirella DePreto's and in her lab, looking at some of these functional networks. And two of them here are the task positive network and the task negative network. And to just summarize these in the simplest way possible, the task positive network is just a network of areas that's active when you have to pay attention to something. And the task negative network, also called the default mode network, is a bunch of regions that tend to shut off when somebody's doing something and tend to come back up into action when somebody's not doing something. And these two networks are anti-correlated. They're oppositely correlated, as you would expect, right? You should be able to go back and forth between attending and focusing to not attending, and those should be very separate networks so that you can clearly separate signal from noise when you need to. And so that's this general approach. Jeff and Mirella DePreto have done a series of studies, and this is one looking at what we call a graph theory approach to connectivity. I'm not going to go into the details of graph theory, but to focus on a couple of key concepts. And that is that as the brain develops, we develop modules, certain areas of the brain which worked together to solve certain tasks. For example, we have a language module in the brain, and there are lots of different language areas, but they all have to work together to make good language function. They have to be well connected with each other, and they have to be separate from other modules. So we call this modularity, and we could diagram it as something like this. And then there are other things as well that we can imagine. How well do these areas within a module cluster together? That is, how close are the connections between regions within a network? And then also, how easily can one network communicate with another network? And we call that path length. Basically, how many connections do you have to go through to get from here to there? And an efficient brain should not have too many connections to get from here to there, right? You should be able to get there as efficiently as possible. And so those kinds of biological networks are known as small world networks, and they're characterized by having hubs of activity that are connected very efficiently with one another. That's a good, nice, normal brain development that has good modularity, that clusters very nicely, but that also can communicate quickly over long distances. So Jeff Rudy, who's now getting the MD portion of his PhD, has looked at some of these values using functional imaging in typically developing children and children with autism. And he looks at this over a wide range of ways of studying it, but the bottom line is that when we look at things like how well these regions cluster together in typically developing kids versus kids with autism, the typically developing kids are showing much more of this clustering together of regions, great communication within a module, and it's much lower in children with autism, significantly so across a whole range of regions. And when we talk about modularity, the extent to which different areas work together in these small little cluster, and the extent to which they're separated from one another with very quick and efficient connections, we find lower modularity in autism and higher modularity in typically developing kids. So these are kids who are more in the 8 to and older range, but this is something that we can also begin now to study in younger children and hopefully look at the whole development of this modularity of function and the communication of function. So this also relates to the concept of neuroplasticity. That is that experience, as Dan has emphasized earlier, experience is what reinforces these functional connections. So while we're born with some functional connections in the first few years of life, we develop lots and lots of them. It's the experience that really solidifies these connections. And over time, what basically happens in the brain is that the brain sacrifices plasticity, the ability to take any of those many routes from the valley down to UCLA, and instead goes for efficiency, that one big old freeway that will get you straight there and fast. And this is something that happens through development. It's the normal developmental trajectory driven by experience. In autism, there appears to be this general pattern of abnormal development of this whole concept of connectivity. And generally what we're seeing is decreased long-range connectivity and an overabundance of short-range disorganized connections. So it would be like driving through little tiny side streets all the way to UCLA, which would take a really long time and wouldn't be very efficient. So the next a couple of studies that I want to just briefly mention is trying to link these concepts of connectivity with autism risk genes. And so I'm going to talk about a couple of the risk genes that Dan just mentioned to you, that particularly this one that he has, that his lab first discovered, the CatNap2 gene. And one of the things that he found was that this gene was expressed in brain in regions that we already knew we found abnormalities in autism when we did functional imaging studies, and particularly the frontal cortex and the striatum. And here are some of those regions on the right. And so one of our former graduate students, Ashley Scott, brought in two groups of children. First, a group of kids with autism and children with age match without autism. And then she replicated the study on a much larger group of children who were just typically developing children. And she ignored autism diagnosis and just asked the question, when we look at areas of the brain that are functionally active and functionally different in autism versus controls, how are these regions connected to the rest of the brain? What is the connectivity pattern in individuals who have the risk gene versus those who do not have the risk gene? And this is what she found. What she found, and it's best summarized in this graph here, that kids who did not carry the autism risk alleles showed stronger connectivity in these long-range connections, and they showed less connectivity in these short-range local connections. Children who carried the risk gene, the autism risk gene, showed decreased long-range connectivity, but increased short-range connectivity. So it was sort of the opposite pattern. But what's really interesting about this was that these are not, we're not comparing children with autism versus those without autism. We're comparing individuals who have the risk gene regardless of diagnosis. And so basically what she found is that having a risk gene for autism biases the brain towards a particular pattern of functional connectivity. It just biases the brain towards more local connectivity and for having not as much long-range connectivity. So it's not a causal gene, and it's not a causal process, but we imagine that if you had enough of this going on, and if this were reinforced enough in an environmental experience, that ultimately that would lead to autism. And this is another gene. Oh, it's in yellow, so you can't see it very well, but this is the MET gene, and we see the exact same phenomenon here, this pattern of decreased and abnormal connectivity in those with a risk gene compared to those without. And this is true for kids with autism and also for kids without autism, although it's a little bit worse for the kids with autism. Then I wanted to show you a different gene. This is the a gene for, a series of genes for the oxytocin receptor, and oxytocin is what Dan just talked to you about in his studies, showing that administration of oxytocin recovered a deficit in some of these knockout models. Oxytocin is very important. There are a lot of different risk alleles that are associated with the oxytocin gene. We call them SNPs, single nucleotide polymorphisms on this autism receptor gene. but they have consistently been associated with social deficits in empathy in older normal volunteers, with deficits in attachment, with autism risk, and also with altered brain structure and function. So for example, this study showed reduced gray matter volume in this area of the brain in those with these autism oxytocin risk genes, and also decreased activity in the amygdala, an emotion center of the brain, for those with a risk gene. So there's good evidence that variability in this oxytocin gene, that's an autism risk gene, also leads to changes in brain structure. Well, what about connectivity? So one of Morella's really terrific now graduate students, Liana Hernandez, has been looking at another brain network called the Salience network, and this is a network that's very important in picking out from the environment what are the most important things in the environment. And one region, particularly this one, the anterior insula, is a hub region in that network. So we're looking for then connectivity between that hub region and other areas of the brain in the network, and the strength of those connections with individuals who have and who do not have this risk gene. And here's what she found. She found that when she took a seed in that hub region in the insula and looked at the connective structure between that region and all the other regions in the brain in that network, what she found was that the individuals who did not have the risk, so non-risk carriers, the GG variant of this gene, had good, strong connectivity in many regions throughout the brain. Those who had one or both copies of the risk gene had much less connectivity from that seed region in the Salience network. This is the difference between carriers or non-carriers, and so you can see that a lot of these are very long-range connections that have reduced connectivity for those carrying the risk gene. And again, this is not just individuals with autism. This is true for carriers of the gene regardless of diagnosis. The other thing that she did, which is very important, was she looked at how many of these different risk snips, as we call, subjects had, and looked at, is there an additive effect? If you have multiple risk genes in this oxytocin receptor gene complex, multiple risk alleles, how does that affect social functioning? How does it affect IQ? And what she found was, so these are all the SNPs that she studied, what she found was that the more oxytocin risk genes a person had, the worse their social responsiveness was on the SRS. And the more of these risk alleles that a person had, the lower their IQ was. And this was true regardless of diagnosis. So, thinking about connectivity and plasticity, in general, there appears to be a broad pattern of abnormal development of connectivity, both structural and functional in individuals with autism. And it seems like these autism risk genes, in general, just bias the brain towards this abnormal pattern of connectivity that emphasizes local connections at the cost of long-range connections. And this is, so we know that experience drives this establishment of functional connections, and as well as the pruning of unnecessary connections. And so in our minds, this reinforces the absolute necessity to identify children as early as possible and intervene as early as possible, because these changes in the brain are experience driven and experience reinforced. So the earlier we can intervene in that developmental trajectory, the more we will hope that we can change that trajectory and make it a more normal trajectory. And that's why we're doing high-risk infant developmental studies and intervention studies. And I just wanted to mention some future directions that we're doing here. We are looking at now super high-risk infants. So these are infants who have a sibling with autism and one other relative with autism, and scanning their brains at 6 weeks of age and following them through the first year of life. And this is some very, very early data that Mirella DiPreto has acquired this from an 8-week-old infant looking at resting state connectivity. And even at 8 weeks of age, you can already see these resting state networks, which is really kind of interesting. And this is a functional activation study where she's compared passive listening of English phrases versus Japanese phrases, which the children were not exposed to in either in utero or early in life, and looking at functional activity and auditory cortex. And I think that you can pretty well see with the bare eye that there's more activity for the infant's own language than there is for the foreign language. This is the difference between the two conditions for this one infant. And this is only at 6 weeks of age. So the brain, even at this very, very early age, is already forming these connections, is already responding to critical information. And so it's never too early to be studying these children. That's a really good question, and it hasn't been studied, but those of us who are doing this kind of work believe that. We believe that that's part of it. I think there are other parts probably as well. We are advocating a model of implicit learning deficit in autism, and we think that individuals with autism focus more on rote learning systems than implicit learning systems. That could also contribute to that. But yes, I think that you're absolutely right. I think that if you spend a lot of time focusing on one small area, you will develop and nurture that area, and you can become better and better at it. And so that very well may underlie those splinter skills, but as of yet, it has not been studied. We haven't studied it, but I know that Pat Levitt's group has studied that recently over at USC, and I'm recalling correctly that he found that kids with a lot of them have digestive problems had delayed language, and that when you improved some of the digestive problems, their language improved as well. It's not really clear exactly how they're linked, but there does appear to be some link with that, which is really interesting. I would like to thank you all and to remind everyone that today's research is tomorrow's treatment, and to strongly encourage you to volunteer for studies, and obviously thank all of my great collaborators here, and thanks for your attention.